Mr. Simpson, look at this lineup and tell us if any of these are the aliens you saw.
Yo!
No, I'm sorry.
Oh, this makes me very angry.
Come, Nerevar, let me give you a lesson in astrobiology. Taken at face value, Martians are not all that different from Earthlings. They are carbon-based, need liquid water, and also require classic elements like nitrogen, hydrogen, oxygen, sulfur, and phosphorus to function. But it is the combinations of these elements that makes them different. For storing information and replicating, Martians use a polymer derived from glycol nucleic acid, although billions of years of evolution made it more complex and perfected it so much that it does not resemble anything we were yet able to synthesize in our laboratories. Functionally, it works almost the same as DNA, though the majority of Martian life forms have 5 rather than 4 nucleotide bases like we do. They also have a simpler, even less well-understood polymer that most likely serves as an analog to RNA. Genetic studies comparing the heredity of the extant life forms to each other have only started in the last few decades. Until then, the reconstruction of the Martian tree of life was largely reliant on anatomical and fossil analysis, which is not always reliable as we have seen in the case of Earth. 2 life forms looking similar to each other does not automatically mean that they are closely related. Comparing embryological development have also been difficult, as humans have struggled with raising these extraterrestrial life forms in captivity. I shall now here list the important divisions of life on Mars, ordered by increasing complexity, though note that this does not indicate a line of descent. First, if there was ever an example of a perfect wastebasket taxon, Xenovera would probably be it. A wastebasket taxon is usually an unnatural taxon into which various unrelated species are lumped in due to misleading similarities and lazy taxonomists. In this case, Xenovera has become a catch-all term for any extraterrestrial biologic molecular structure that, as the name implies, even slightly resembles a virus. And by any, I do mean any, as even similar structures found in Jupiter's moon Europa and Venus's atmosphere have been called Xenovera. They are all characterized by being too simple to be, by most definitions, considered life, but still showing enough reproductive and Darwinistic capabilities to be in some kind of prebiotic limbo. Another defining trait is that all of them are exclusively parasitic, preying on and abusing the metabolisms of actual life forms. But that is really where the similarities end. Some resemble classic viruses from Earth by being genome strands encased in membranes or protein shells, while others are just free-floating strands of biopolymers or even just self-replicating proteins, similar to prions like mad cow disease. Some may be survivors of an ancient prebiotic primordial soup that evolved to prey on later life forms, while others may be the descendants of actual life that lost complexity over time as an adaptation to parasitism. Due to this heterogeneity, many astrobiologists have tried breaking up Xenovera into multiple natural groupings. The true viruses of Mars, which possess genetic material similar to the other Martian life forms and capsids, are called Xenovera or Ariovera. They are predictably the most common pathogen in the Martian biosphere, though they are thankfully not capable of infecting humans, as our DNA and cellular systems are incompatible with theirs. The same is true for any other Martian pathogens, as well as terrestrial ones in reverse. But handling foreign microbes or introducing our own is still considered dangerous for both parties. Even if we cannot infect each other, some microorganisms might still release waste products that are unexpectedly toxic to other life forms. Next up are the Nanobacilli, informally also known as nanobacteria. This taxon has no real equivalent to Earth life. Originally grouped among the Xenovera under the name nanoglobuli, they were long thought to be too simple and primitive to be considered life as we know it. But this changed as it was discovered that they met all criteria to be considered life forms. Unlike viruses, they are capable of independent reproduction and metabolism, despite most of them being smaller than 200 nanometers. This is rather challenging, as the existence of bacteria in this size range was previously deemed impossible, since the bodies of such organisms were considered too small to hold all the necessary molecular machinery for independent replication. This may have been true for terrestrial biochemistry, but apparently things work differently on Mars. By some poorly understood process, the nanobacilli seem to have adapted an internal machinery that consists of protein-based alternatives to the genome used by most other Martian life, allowing them a rudimentary metabolism and reproduction without having classic genetic material. Yet they are all encased within membrane structures closely matching those of Ariants, making it possible that they descend from more complex cells that have simplified to an extreme degree. In a sense, they are a bacterium trying to be a prion. Nanobacilli can live in a variety of lifestyles and habitats, though autotrophy is relatively rare. Commensalism with more complex organisms is instead the most common way of life for these organisms. Arianta are the closest analogues on Mars to earthly prokaryotes. Although still single-celled, they are usually far larger and more complex than nanobacilli. They either have heterotrophic or autotrophic lifestyles, reproduce asexually, are one of the major backbones of every ecosystem, make up the majority of Martian biomass, and have probably ruled this planet for over 4 billion years. Just your average bacteria. In an interesting contrast to terrestrial prokaryotes, Ariant cells have a significantly higher propensity to evolve intercellular membranes, meaning walls and tendrils that extend into the cytoplasm. These internal membrane extensions function more similarly to the endoplasmatic reticuli of Earth's eukaryotes and often serve as workspaces for enzymes and proteins. Why Martian prokaryotes have a higher tendency to evolve such intercellular membranes than Terran prokaryotes is not known. This might simply be a consequence of differing biochemistries. All higher life forms on Mars, and likely even the nanobacilli, are descendants of the Arianta. The kingdom could therefore be considered a paraphyletic and therefore unnatural clade, as it does not include all of its descendants. Important phyla include the Phytoarianta, the Methanoarianta, and the Percloarianta. Next up are the Macroarianta, which are what one could call a multicellular prokaryote, as they are not just simple cell colonies, but complex organisms made up of differentiated cells. But their individual cells are still structurally Ariants. A multicellular bacterium does indeed sound strange, but there are similar life forms even on Earth, such as the prokaryotic myxobacteria and several species of cyanobacteria that have multicellular life stages, complete with cell differentiation and fruiting bodies. Macroarianta are similar to these, although far more widespread on the red planet and more complex, some resembling microscopic versions of plants, fungi, and even animals. Despite this complexity, none of the known species grow larger than 15 centimeters. Some larger fossil relatives possibly show that this was not always the size limit. If this is true, modern members instead seem to have shrunk due to competition with Rhodocarya. Rhodocarya have on average much larger cells than Macroarianta, meaning that a macroariant of the same size as a multicellular rhodocaryote is at a significant disadvantage, since its body is made up of many more cells, making it more prone to cancer and similar issues. Macroarianta seems to be polyphyletic, as apparently not all of its members have the same common ancestor. Rather, several lineages of Arianta evolved multicellularity independently from each other. The Macroarianta were long thought to be transitional forms between Arianta and Rhodocarya, but this seems increasingly unlikely, as Macroarianta lack camaculae and the ancestral Rhodocaryotes seem to have been unicellular. Some extant photosynthetic species build shells made of silicon, similar to diatoms. Entire sediments made of these silicon shells have been found in marine fossil assemblages, showing that Macroarianta once constituted a major part in ancient planktonic communities, making them important for biostratigraphy. The clade of the phyllolithophores are today also among the most important nitrogen fixers. Rhodocaryotes are characterized by their cells having something akin to mitochondria, in their case called camaculae, that engage in oxygenic respiration, as well as other organelles, which is why they are considered the Martian analog to Earth's eukaryotes. They encompass many taxonomic kingdoms that come in unicellular, multicellular, and intermediate forms, and constitute the majority of the macroscopic biosphere, though they have lost a lot of their former glory. It was originally thought that their organelles evolved out of Gigabacilli, the largest known nanobacteria, originating through an endosymbiotic event where an Ariant cell was permanently inhabited by a nanobacillus, either through symbiosis or perhaps even through parasitism. The parallels of this idea to Earth's eukaryogenesis, where an archaean cell was entered by a bacterium, are obvious and might reflect some form of chauvinism by earlier researchers. But recent research has cast significant doubt on this hypothesis. The camaculae bear no resemblance to nanobacilli, do not float freely inside the cytoplasm, have no genetic material of their own, and are not shared between cells during reproduction. Instead, they develop directly out of the inside of the cell membrane after mitosis. The more likely scenario is that, as Mars gradually built up more oxygen in its atmosphere, the ancestral hydrogenotrophic Arianta cells began to protect their anaerobic methanogen metabolism by folding their intercellular membranes onto themselves to create a separate chamber inside the cell. The protein-studded walls of the resulting chamber could then effectively filter out free oxygen to create a controlled anoxic environment inside the chamber, while the rest of the cell and the environment were oxygenated. Afterwards, aerobic respiration may have originally evolved to more effectively eliminate free oxygen from the cell before this gradually became the main metabolism. What corroborates this are recent genetic studies which show that rhodocaryotes, which only engage in hydrogen respiration, are actually more ancient than oxygen-respiring ones, and not the other way around, as originally thought by analogy to Earth. The fact that life on Mars evolved the complex cell completely autonomously and not through an accident like Earth's eukaryotes seems to have had a tremendous effect on the story of the biosphere. Biogeochemical signs of rhodocaryotes show up in remarkably ancient rocks 3.4 billion years ago, far older than the first signs of eukaryotes on Earth, and as fossils show, this seems to have facilitated the evolution of complex multicellular life only about 400 million years later. Next are the Protoroario zoa, which is technically another paraphyletic taxon that encompasses several kingdoms of unicellular rhodocaryotes. Far too many to mention them all here. The many lineages of Protoroario zoa can be distinguished from each other by their modes of life, cell structure, and the way they move using their flagella. All rhodocaryotes ancestrally reproduce sexually, although asexual reproduction has independently evolved in multiple lineages. A prominent group of Protoroariozoans are the flectoids, a semi-multicellular group which resembles a mix between slime molds and lichen. Aerophyta is the kingdom that includes large, multicellular photosynthetic rhodocaryotes that share one common ancestor. They are in some ways the Martian analog to plants and algae by having cells with cell walls and being immobile autotrophs. However, they are not actually the dominant constituents of the global flora. They originally seem to have been during the Thermozoic era, but have gradually declined in favor of Spongesporia and Fractaria. A plausible explanation for this might be their metabolism. Aerophyta do not engage in oxygenic photosynthesis like Earth plants, but instead use sunlight to react hydrogen sulfide with carbon dioxide to generate sugar, and as a waste product, elemental sulfur. This is a form of photosynthesis only used on Earth by some of the most archaic organisms. Aerophytes are of a principally two-layered build, with an external germ layer consisting of oxygen-respiring cells, which protect an inner layer of anaerobic cells that are specialized for this mode of photosynthesis. Such a metabolism seems to have been viable even on land in the earlier periods of Mars, as constant volcanic outgassing continuously enriched the atmosphere with sulfuric gases. But as volcanic activity died down, the Aerophytes lost much of their livelihood, and the increasing oxygenation likely made it even more difficult to maintain their metabolism. Today, Aerophyta without special adaptations, such as the divisions Porphyta, Cochleophyta, and Arechlorotia, are solely found close to the hangs of active volcanoes or along geothermal hot springs. More derived groups have gained a more flexible range by living in endosymbiosis with sulfur-reducing Ariants, which recycle the waste sulfur back into hydrogen sulfide. Part of this group are the Arthrophyta, which are notable for possessing a segmented body with both internal and external bilateral symmetry, something not seen in any living plant on Earth, though it reminds me of some flora on Nern. Lastly, there are also the Pentatophyta, possible offshoots of the Arthrophyta, which have lost their autotrophic lifestyle and have instead become heterotroph detrivores, fulfilling a similar ecological role on Mars as fungi do on Earth. Unlike Earth plants, Aerophyta have no alternating generations. Like the Aerophyta, Spongesporians possess cell walls, though composed of different polysaccharids than in Aerophytes. Spongesporia have no true differentiated tissues and they are immobile their whole life. But since they are heterotrophic, they are not reliant on sunlight or chemosynthesis. These traits combined with their rhizome networks makes it easy to draw parallels between these life forms and Earth's fungi. But the similarities are superficial. Fungi can exist both in unicellular and multicellular states and are mostly made up of loosely connected hyphal networks without a central body. Spongesporia, on the other hand, are exclusively multicellular and have a more defined morphology. They have a central, often tube-like trunk and a root-like network of hyphae underneath. The earliest members of this group seem to have lived on the seafloor, had porous walls with a hollow central cavity, a sclerite skeleton made of silicon dioxide, and ate by filtering the seawater with cilia. This made the earliest Spongesporians, as the name implies, more similar to Earth's sponges, and indeed many extant members still have the characteristic porous body cavities and sclerite skeletons. Consequently, the majority of Spongesporians live as filter feeders, not just underwater in cave systems and glacial lakes, but also on land, where the low gravity allows for the existence of an aeroplanktonic community. Some forms also live in symbiosis with photosynthetic Ariants, essentially being giant sponge lichen. Spongesporia reproduce through spores and have haploid and diploid generations, which often differ greatly from each other. Fractaria are a lot more complex than Spongesporia and Aerophyta, with clearly visible symmetrical builds and differentiated tissues, but they are still too primitive to be comparable to animals. The majority lack a gut, mouths, or any digestive organs, as well as the cell walls and hyphal roots of the Spongesporians. While they ancestrally were filter feeders like the Spongesporia, many have also become autotrophs by living in symbiosis with colonies of Arianta that live in their tissues. These forms live off osmotrophy, chemotrophy, and even photosynthesis. Because they are relatively easy to keep in captivity, their biology has been well researched. A Fractarian starts out life as an immobile propagule which grows into a base not unlike that of a Spongesporian embryo. Out of this, a fern-like frond develops with a simple internal system of interconnected filtering chambers. This frond gives rise to another frond, which also gives rise to another one, until the whole organism ends up with the characteristic glide symmetry that the group derives its name from. Such basic fronds seem to be the simple building block of this kingdom, with most members having found some way of iterating in remarkable ways on this simple template, for example by evolving multiple veins or increasingly more fractal fronds. The majority of the higher Fractaria, however, reach their complexity instead through colonial living, like corals, sea pens, or sessile versions of the Portuguese man-o'-war. Many clones growing from a single ancestor grow into larger, plant-like superorganisms. An important group among these colonial forms are the Hylozoa, which have evolved outer walls made of cellulose, similar to wood, which allowed them to colonize terrestrial habitats. Through symbiosis with Phytoarianta, they have become a major part of Martian flora, in the past even growing into forests. Fractaria are generally not classified as Martian animals, since at no point in their lives do they have a mobile stage. The sole exception to this, as has only recently turned out, is the superphylum Pseudarticulata. Originally classified as primitive Arazoans, we now know that these are actually Fractarians, which evolved from solitary fronds that found a way to use the filtering cilia on their skin to lie flat on the ancient seafloor and crawl about the microbial mats like giant amoeba. Out of these primitive forms eventually derived descendants, which independently from Martian animals proper, evolved their own versions of nervous systems, muscles, and sense organs. Pseudarticulata, with their pseudo-segmented body, today largely resemble flattened worms and maybe arthropods. Their most successful and famous clade are the Chirorbita, sometimes nicknamed Martian trilobites, which have evolved a tunicine exoskeleton, camera eyes, and move about with hydraulic tube feet similar to starfish. At last comes the kingdom Arazoa, which is most comparable to the Animalia on Earth, and shall be referred to as animals from here on for simplicity's sake, even if it is not entirely correct. They have differentiated tissues, lack cell walls, are heterotrophic, and are all capable of free movement at least at some point in their life cycle. Interestingly, though, the majority of them possess metabolic pathways that are capable of recycling much nitrogen from their waste, which makes them more similar to earth plants than to earth animals. The exact number of Arizoan phyla that exist or have existed on Mars is a debated question. Instead of discussing each in detail here, you will get to know them by example in the upcoming videos, perhaps with help from a friend of mine. The exact origin of the Arizoa is a controversial issue as we lack a lot of fossil or embryological data. Anatomical comparisons predict that the Fractaria are their closest relatives, but researchers have also noted similarities to the Spongisporia. It is interesting to note that unlike most animals on Earth, the Arizoans are predominantly hermaphroditic, just like that fucking imbecile Vivek. During mating, either both partners get pregnant or, what more often happens in the more derived groups, the partners first fight each other to determine who gets to impregnate the other, something we might want to reenact, Nerevar. Many of the simpler forms also practice self-fertilization, similar to Earth plants. Why this difference exists is unknown. There are hypotheses whereby the difficult inheritance of mitochondria between cells is what drove sex differentiation in Earth's eukaryotes. Since Rhodocaryotes do not have true mitochondria, the true need for sexual differentiation may have never arisen on Mars. I hope you enjoyed this video and look forward to the coming ones. Make sure to like and subscribe, visit the project's original website, and maybe also check out my Patreon, Unwa.